Hello everyone, welcome to the next lecture in the lecture series of fruits and vegetable processing. Today we will be studying about plant growth regulators. So under plant growth regulators, first let's see what is plant growth regulators. So as per definition, so these regulators, these are substances used for controlling and modifying plant growth. Okay, so these either control or they modify, like it can be negative, in a negative way also, that they inhibit the growth, or they modify, that means they can increase the growth also. We will study about it in detail later. So these are the substances used for controlling and modifying plant growth processes without appreciable phytotoxic effect, that is, it should not exert any toxic effect on the growth or the health of the plant. Okay, that is, it should not be toxic to the plant at the dosage levels, that is, at the dosage level, at the in the amounts in which it is applied to the plant, it should not exert any toxicity on the plant, right. So the naturally occurring endogenous, or that is the naturally occurring growth substances or the endogenous substances, these are called your plant hormones, while the synthetically made these plant growth regulators, these are known as growth regulators. Okay. So the synthetic ones are your growth regulators, while your natural ones or endogenous ones that are produced by the plant itself are called your plant hormones. So now these growth regulators which are synthetically made, they essentially mimic the action of your natural ones. Okay. The function of the both of these are same, like if you say AUXIN is an example of plant growth regulator. It is both naturally produced also, then it can be synthetically made also to use externally in case of if your plant is not producing adequate amount of your plant growth regulator. So you can apply it artificially or manually on the plant so that the growth can be initiated in that case. Okay. So this is your plant growth regulator. They modify or control the growth processes without any toxicity at the applied level, right. So next we see the plant hormone is an organic compound. So we talked about endogenous growth regulators or endogenous modifiers are called your plant hormones. So this plant hormone, it is an organic compound synthesized in one part of the plant and translocated into other, that is, it is produced by one part of the plant and then it is sent to the other part to exert its effect on that part. Suppose, okay, so translocated to the other part where in very low concentration it causes a physiological response, that is, so these are sort of hormones, right. So as in case of humans, these hormones, they are produced in a one place and they are sent to other part of the body, or in this case other part of the plant to exert their physiological effect. Now these, since they are hormones, so these are required in very low quantity and to show their effect. Okay. Now the plant hormones are identified as promoters. So under promoters, that is, they promote or they amplify the growth. So under promoters we have auxin, gibberellin, cytokinins. Okay. So these are just some examples of plant growth promoters. And then we have inhibitors, that is, they will inhibit or they will nullify or they will hinder the growth of the plant in one way or the another. Now under inhibitors we have abscisic acid and ethylene. See, ethylene you have studied, it is helpful in ripening. Now there is ethylene, it performs both function. It can be used as a promoter also, or it can also be used as an inhibitor. Suppose you require ripening, so in that case it will suppose you require ripening. So in that case ethylene will serve the purpose of a promoter, while in other case, suppose if ethylene is applied on the roots, so it will inhibit or it will hinder the growth of the roots. So in that case it is serving as a inhibitor. Okay. Now there are other growth substances also, like besides this inhibitor and promoters we have other substances like florigin, flowering hormones, some others are also there like domins. So these are under other substances. So they are like flowering agents. So these flowering agents, they are sprayed onto the plants in order to promote flowering of the plant. Now there are other substances also which can inhibit flowering also. Okay. So these are the categories: promoters, inhibitor. Then we have a third class, other growth substances. So we will read about all of these in detail. Okay. Now under classification, if we see, so under classification, your PGRs or plant growth regulators, they can be divided into these general categories. Okay. So under these general categories, your PGRs are divided, and they are based on their chemical composition or the effect exerted by them. So first you have auxin, then gibberellin, cytokinins, ethylene, dominins. See, you first four in if you see generally, so you categorize them as 5 categories. Out of those 5, 4 are these: auxin, gibberellin, cytokinins, and then you have ethylene. All other growth regulators or all other growth substances, so these are categorized under the category other substances. Okay. So basically we will study about these 4 and then other category or other growth promoters or growth substances. Okay. Now under auxins we have some example given here: IAA, gibberellin, GA. So we will read about all of these in detail now. So let's move on to the next one. So first of all, auxin. So this is your first category, auxins. So auxin, it is a chemical substance, okay, chemical substance that stimulates cell division or elongation, stimulating a plant stem to grow taller in a specific direction. That is what is the main action of auxin is. So it will promote the stem of the plant to grow in upward direction. It will prevent spreading of the branches. That is, your stem, main growth will be in the main stem of the plant, while your secondary branches or the secondary buds that are coming out of it will be suppressed. Okay. So under auxin we see the characteristic feature is polar translocation. That is, auxin based on the movement of sun. So this can bend the plant, like upward movement of the plant can be bent. In case of suppose if auxin is applied on the stem, so stem will move upward, it will direct it to move only upward, while if it is located or if it is applied on the roots, so roots will grow downward. So auxin will control the movement of growth of the plant. Okay. Then we have apical dominance. Under apical dominance, what happens is, suppose your plant is growing upward, right? This is the main stem. So and these are the roots of the plant. Okay. So what happens is, your branches will grow out of it, right? And then these branches will further bear leaves. Okay. But what happens is, in case of we are applying auxin, so this will only promote the growth of your main stem. Okay. It will promote the growth of main stem, while your secondary branches will not emerge out of it. Okay. Suppose your secondary branch was supposed to emerge out, so it will be suppressed, right? So this is your apical dominance. Okay. So again, variable behavior of root and shoot growth. Okay. So on root as well as on your shoot growth, your auxin has a different effect, like shoot will grow upward while your root will grow downward. Suppose if after application of auxin you place your plant horizontal, so what will happen if you are placing your plant horizontal, this is the stem suppose, okay, so and this is the roots. What will happen after application of auxin? This stem or this main stem will start to bend upward, it will grow upward, while your roots will start growing downwards. Okay. So this is what happens when you apply auxin. Then it has variable behavior on root and shoot growth. Okay. It also initiates root growth when you apply auxin. So your root growth will be initiated, that is, more roots will be formed, it will be more bulkier or more branched than your normal roots. Okay. Delay in abscission and differentiation of xylem element, that is, the abscission you have studied under ripening, as the fruit ripens, abscission point is formed, your that abscission starts to dry out. So it delays that abscission phenomena, so that there is more regular supply of your nutrients and your food material to the fruit. Okay. Your maturity will not be reached so early. Okay. And differentiation of xylem elements. So auxin has all these characteristics. Further we will study about its uses. So as you can see here, in this case, see, this is your normal stem, and when you apply auxin is produced here. Okay. So it will start to move, it will start to bend. Okay. in the direction of your light, right. So this is how auxin works. Next we will see uses of auxin. Okay. Under uses of auxin we will see here propagation. So under uses of auxin we have propagation of plant by hormone treatment of cutting. Suppose if you are propagating your plant, that is you are grafting your plant, your part of your plant to other place. So application of auxin can help in propagation. If we apply this hormone onto that plant, so that will not die out easily, and there are more chances of survival of that part of the plant or that part of the cutting. Okay. So it can help in propagation of the plant. Then prevention of your pre-harvest drop of plants, that is before harvest your plant, it becomes heavy, right, the stem, it will start to bend out. Okay. So your auxin application will prevent that bending out or drop of your plants, right. So next, your auxin helps in increasing parthenocarpy, that is your fruit development will start before pollination or before fertilization of the ovary of the flower. Okay. So that will result in your seedless fruits. Okay. So parthenocarpy, it increases development of fruit before pollination. Then we have increasing fruit set, that is from one branch of the like fruit grows at a particular point, right. So it will be like number of fruits growing out from one point. So it increases fruit set from a single plant. Then prevention of sprouting of by initiating buds, that is sprouting will not occur, buds like it will initiate, sorry, it will inhibit the buds. So as we have studied earlier, it promotes stem growth, right, the main stem will grow, the buds will be inhibited. So further like branching will not take place, right. So these are the uses. Then again we see inhibition of prolonged dormancy, it prevents prolonged dormancy of your plant, that is it will initiate growth. If your plant has been dormant, it has not been showing signs of growth, so it will prevent that dormancy and will push the plant in a growth period. Okay. Then control of flowering. Okay. It will control flowering, it will sort of slow down the flowering process, or less number of flowers will be there. Okay. Then we have defoliation of plants. So under defoliation of plant, so it prevents defoliation, that is your leaves will be formed on the single stem. Okay. So then is prevention of leaf fall and abscission. So it prevents leaf fall, that is the abscission, the point at which the fruit is attached to the tree, so that dries out and your fruit falls. Similarly for leaves also, so it will prevent that leaf fall and abscission, right. Now thinning of compact fruits and selective weed killer. So these are the uses of auxin. It also like serves as a weed killer. Okay. So any unwanted plant, if with the application of auxin, it can prevent the growth of those weeds. Okay. Then we'll read about gibberellins. So your second plant growth regulator is gibberellin. So under gibberellin, it is the second most important growth hormone. So there are more than 60 types of gibberellin present in your plants. So these are named as GA1, GA2 up to G60. Okay. So there are more around 60 gibberellins, and about 51 types are found in higher plants. So gibberellin, what is the function as a mechanism of gibberellin? GA exerts, or gibberellin exerts its physiological effect on altering the auxin status of the tissue. That is, it will alter the auxin status. Auxin is naturally produced by the plant. Okay. So it will alter the auxin status if it acts at the time at a gene level to cause depression of specific gene. So gibberellin acts as a specific gene level, and it prevents or it depresses a specific gene. So it is like it will if a particular gene is suppressed, so it will not show its effect. So certain effects will be absent. So we can selectively identify the gene which we want to suppress, or whose effect we do not want to see in our plant or in our fruits. So we can select that, we can target that using gibberellin, and we can prevent that effect in your fruits or plants, right. Again, it will also gibberellin also promotes growth of your plant here. So here you can see your upward movement, or like your plant will become more taller. okay, by the application of your gibberellins. So, applications of gibberellin, it increases germination. So, if it is applied on seeds, so it increases the germination of the seeds, inhibits root growth, leaf expansion occurs, okay, your leaf will become more large. So, in case of your cabbage or sweet corn, so cabbage is all leafy, right. So, if the leaf size is enhanced, so your cabbage will be of a very huge size, the size of the cabbage will increase, hence your produce. Then again hyponesty of leaves. So, your hyponesty means the lower part of your plant will become more pronounced, that is the more growth will be there in the lower part of the leaves or the lower part of the plant. The upper part, so that a straight movement is observed, the plant will not bend out easily. Okay. It induces flowering in long day plants. So, it can cause plants to flower in long day plants. Then increased fruit setting, that is your fruits will set earlier, or it will enhance, fruit setting will enhance. Then stem elongation, we have seen in the previous slide, your stem will elongate. Stem elongation with increased number of internodes. Then same with the number of internodes in the stem will increase with the application of your gibberellin. Breaking dormancy of plant buds. So, if your plant buds have been dormant for a specific time, so with the application of gibberellin, your plant will start to grow out, that is these buds, they help to branch out the plant, right. So, more branches will be produced, it inhibits or it breaks that dormancy and supports the growth of plant later, like branching will be promoted. Then if sprayed on fruits, it is sprayed on fruits to prevent rind disorder. So, rind disorder is, as you can see in this picture of oranges, so these are your rind disorder, that certain patches will be observed on your plants or your fruits, particularly fruits in this case. So, certain patches will be observed in your oranges. Suppose in this case we are taking the example of orange. So, these patches with the application of gibberellin, so these rind disorder will be prevented, you will not get these patches. Again we have seen, right, fruit setting will be enhanced or branching will be more. So, in case of grapes, this is without application of gibberellin, that is it is untreated, and this one is treated with your gibberellin. Okay. So, with treatment with gibberellin, so your branching will be enhanced. So, in case of grapes you will get a more bulkier this bunch, while in untreated one it will be elongated. Okay. So, this is your plant growth hormone gibberellin. Next, let us see. Next one is your cytokinins. Under cytokinins, cytokinin affect cytokinesis, that is your cell division. So, cell division, if it enhances cell division, so what will happen is your growth of your plant will be enhanced in every direction, not only up but otherwise also it will start to branch out. Okay. It affects the cytokinesis, it promotes cytokinesis or cell division or cell enlargement. Cell division will be enhanced, your cell will become large, it inhibits dormancy, flowering will be enhanced, fruit setting will be more, you will get more fruit sets, and it delays senescence. Okay. In agricultural application, this translates into propagating new plants in the nursery. Okay. If in a nursery we see, so the cytokinins, these are helpful in propagating new plants, like from one plant if you are grafting or if you are cutting one part of the plant and sowing it somewhere else. So, with the application of cytokinins, as it helps promote cell division, so your plant will grow further, like it will grow fast, there are less chances of plant dying out during propagation. Okay. Breaking seed dormancy, if your seeds are dormant, they are not growing, so with the application of this cytokinin, so your seeds will start to germinate. And delaying senescence, so senescence is delayed in case of fruits, your fruits will spoil later as well as plants also. With the application of cytokinins, fruits or your plants will start to senescence or die out later at a later stage. Then initiation of cell division, delay of senescence using tissue culture. Tissue culture I already told you, as it promotes cell division. So, it will help in growing the tissue faster. Counteracts apical dominance. See, in apical dominance we studied that there will be main stem which will grow upward, and there will be no branching of plant take place. But in case of cytokinins, as you can see in this picture, suppose this was your normal plant after application of your auxin or any other plant growth hormone which promote only upward movement or growth of the stem without branching. So, with the help of cytokinins, you will get a more full plant, that is branching will be enhanced and you will get a lot of branches. It promotes branching of your plant, okay, or if your dormant buds are there, it will promote their growth, right. So, this is how cytokinins work. Next we will see about uses or application of cytokinins. So, cytokinins, they are helpful in cell division, cell enlargement, we have already studied, morphogenesis, dormancy, it breaks the dormancy of the plant. Then apical dominance means it counteracts, okay, apical dominance will be prevented, dormancy will also be prevented, that is it will initiate growth, okay. Then mobility, immobile, it is immobile, obstructs the movement of amino acid, phosphates and various other substances, okay. Then nucleic acid metabolism, quick increase in the amount of RNA and decrease in the DNA. So, your RNA will be enhanced. So, RNA if RNA is enhanced, so your more parts of the plant will be formed, that is it will promote growth or branching of the plant. Then we have protein synthesis, which results in increasing of DNA, and again at an increased rate your DNA will be formed. Then we have florescence, it can also help or it can also function as a florescent, that is promote the growth of flowers, that is induction of flowering in short day plants, that is it will introduce or your flowers will begin to form at a faster rate in your plant with the application of cytokinins. So, you can see here in this case it prevents senescence. So, this is an untreated sample, there is no cytokinins here, simply water. So, if you dip your leaf in your normal water untreated one and the one treated with cytokinins, so your senescence will be delayed. See, in this case your leaf has started to die out, okay, the chlorophyll is already degrading, but with the application of cytokinins your chlorophyll degradation also is delayed, or your leaf will not die out so easily, right. Again with the application on roots, if we apply cytokinins on root, so root will be more branched. You can see here in both the cases, this one is untreated, this is treated. So, at see, these roots are formed after your plant has grown too much, okay, after a particular level of growth. So, your roots will be this much large, but when you apply cytokinins at an initial stage of the plant, so your roots will start to form, it will start to branch out and you will get a bulkier roots, like more amount of roots with the application of cytokinins at an earlier stage itself, okay. So, if you apply in plants like carrot, okay, so if you apply in a carrot plant, so your carrots will be formed, will be bulkier, or it will be very large in size at the initial growth stage only, okay. So, it will promote the growth of carrot or other other fruits and vegetable which are grown from the roots, right. Next we will see ethylene. See, we have studied ethylene earlier itself. So, ethylene helps in ripening. It is a plant hormone, you can say, which helps in ripening. Now, ethylene, it can serve both as a promoter or it can also serve as an inhibitor. Okay, we will study about this now. So, it is highly used in inducing fruit ripening. So, when you are using it for purposes like ripening, so it helps as a promoter. Ethylene is a natural product of ripening fruit. It is a gas at temperature under which a plant can live. So, ethylene, suppose at normal temperature or at temperature in which plant survives. So, ethylene is mostly formed in is present in form of a gas. Okay. So, ethylene affects growth, ripening and senescence in plants. So, auxin increase ethylene level in plant, and auxin actions are attributed through ethylene, such as increased percentage of female flowers, apical dominance, apical bud dominance and leaf apinesty. Okay. So, again that leaf will be more, lower surface of the leaves will be more pronounced, right. Okay. You can see here, so auxin increase ethylene level in plant. So, auxin and ethylene, they are interrelated. So, it will increase the level of ethylene in plant, and that's how it exerts its effect. It will increase the percentage of female flower, and apical bud dominance will be observed, and leaf epinasty will be observed. Then in agriculture application, it is used primarily to promote ripening. So, main application for farmers of ethylene is to promote ripening, although it can also be used both as a promoter as an inhibitor also if ethylene is applied on the roots. So, it will prevent the growth of roots, or it will inhibit up to a certain level. Okay, so, but the main use of ethylene is plant ripening, or sorry, fruit ripening. So, these are in no ethylene has not been treated on this, and third one are treated with ethylene. So, in case of tomato, here no ripening is there, or here those fruits have been ripened under same time period. Okay. Again, like if you keep apple with the avocado, this is just a practical example. So, your avocado will ripen faster because, so apple serves as a source of ethylene. It will secrete, or it will like promote growth of, if ethylene is secreted by apple, it will promote the ripening of your avocado at a faster rate, right. So, again next we will see application of ethylene. So, abscission. So, when with the application of ethylene, your abscission is fastened, or it accelerates abscission. Principal accelerator of abscission capable of promoting changes associated with pre-abscission and aging of leaves, petioles and flowers and fruits. Ethylene degreening is commercial practice, that is your ethylene is applied for degreening of many of your fruits and vegetables. Okay. Up to concentration of 5 to 10 ppm, ethylene will show its effect, and ripening will be pronounced or accelerated, and degreening also will occur, as in case of papaya. So, papaya when harvested is the green in color. So, these can be artificially ripened by application of ethylene. Okay. Then degreening occurs after ethylene treated fruits are exposed to air. Air accelerates maturity and induces uniform ripening, that is after application of ethylene. Application can be applied on this fruit in form of sprays by mixing it in certain chemical, and after application of ethylene, you have to expose the fruit to the air, so that your degreening or the ripening will be accelerated in that case. Okay. So, this is the application of ethylene. Then under other category we have growth retardants. Okay, what is the growth retardants? These are just the function opposite to that of growth promoters. The term growth retarding or growth retardants is that the chemical slowed cell division. Okay, the chemical after application, it will slow the cell division and cell elongation of shoot tissue, and regulates plant height physiologically without formative effects. That is, it will inhibit the growth of the plant in all the cases, like cell division will be inhibited, it will be slowed down, cell elongation will be slowed down, the growth of the plant will be slowed down, it will not gain enough height, the plant will not be of enough height, and again you can see it regulates plant height without any formative effects, that is you will not get any defects also in the plant, or formative effects, that your fruits will not be formed so earlier also without affecting any formation in the plant. Again these are the examples: AMO 1618, then phosphon D, CCC, MC and LR. So, these are the examples of your growth retardants. Then these do not occur naturally in the plant and act in retardation of stem elongation, that is they are not produced by plants naturally. These are only synthetically made and then applied if you want to delay the growth of your plant, or it has certain other application also, like if you apply these growth retardants for weeds. So, weeds will not be grown, it will retard the growth of weeds also, preventing cell division. Okay. So, these are the application of the mechanism, you can say, by which it will inhibit the growth of the plant. So, just an example here. So, this is untreated sample, your plant has grown to a very good height, but after application of your plant retardant, one example is dicite. So, your plant will be retarded, the growth will not be occurring so much. Okay. Flowering will also be affected, and it will not be more pronounced flowering, right. So, next we will see here, next one is inhibitors. So under inhibitors we can see here, inhibitor also suppresses the growth of the plant. In this case there are phenolic inhibitors, synthetic inhibitors and abscisic acid, that is ABA. So we will read about this abscisic acid, but inhibitors, these are of phenolic type. Then we have synthetic. Phenolic ones contain phenol group, your synthetic ones are artificially made, or these may be certain other chemicals which will prevent the growth of the plant. Now phenolic inhibitor are benzoic acid. Under phenolic inhibitor we have benzoic acid, salicylic acid, coumarin and chlorogenic acid. So these are the examples of your phenolic inhibitors. Under synthetic one we have malicardazide, then we have triodo and benzoic acid. So these are the examples of your synthetic ones. Again, then under inhibitors, again they will suppress the growth of the plant, they will affect cell growth, cell division, cell elongation, stem growth. So using them, utilizing various mechanisms, your inhibitors will suppress the growth of your plant. Also they can also suppress the growth of your roots also. Okay, so this is just an example. These are the controls in first case. Then after application, your growth will be retarded. It will not branch out much. Your leaf size may also be affected. It will not be so much, right? Then we have under other category abscisic acid. So abscisic acid generally inhibits growth and germination and promotes dormancy. That is, it will prevent the growth, germination, it will promote dormancy, that is your buds will not sprout, your seeds will also not like seeds are a different case, but in case of plant it will not branch out so easily, your buds will be dormant, your fruits will be dormant. So this is the role of abscisic acid, to stop elongation, induce dormancy, delay germination, inhibit growth processes. So under these four, you can say dimension, it will show its effect. Your cell division will also be hampered, cell division will also be slowed down. So this is the role of abscisic acid as a inhibitor, or you can say as a dormant. That is, it transfers the plant into dormancy. Then under summary we can see here. So in this table, all these plant growth regulators have been summarized. Your gibberellins, auxin, cytokinins, ethylene and abscisic acid. So how they affect the various parts of the plant. See here, germination. So gibberellin will promote germination, it will enhance, accelerate germination process. So others also, auxin, cytokinin, ethylene, they will not affect germination. While growth of to maturity, that is growth of the plant, overall growth of the plant, like gibberellin, auxin, cytokinin, they will promote growth of the plant both laterally or that is branching out, and height will be affected in one way or the other. But your ethylene and abscisic acid, they do not have any effect on the growth of the plant. Then flowering, your gibberellin, auxin and cytokinin, ethylene, they will affect the flowering process, but your abscisic acid has no effect on your flowering process. Fruit development, again with the exception of abscisic acid, all these four plant growth regulators, they are going to affect the fruit development also. Now abscission, that is your drying out of your stem. Except the first three, ethylene and abscisic acid, they will promote abscission, that is in case of ethylene, when ethylene is applied on the fruit, so abscission will be formed at the area where your fruit is attached to the plant. Okay, so it will dry out and your fruit will fall. Similar for the leaves also, with the application of ethylene, your leaves will start to fall out faster, it will accelerate falling out of the leaves. Then seed dormancy, except your abscisic acid, only abscisic acid will promote seed dormancy because it is a dormant, so it will prevent growth. Except these, all these three do not affect seed dormancy, but they have, in fact they have opposite effect. Okay, your cytokinins, your auxin, your gibberellin will promote seed growth. Okay, they will like break the dormancy of the seed and your seed will start to grow, but your abscisic acid, it will induce seed dormancy, okay, prevent the growth of seed. Then we have some toxic effects of plant growth regulators. So under toxic effect, we have in general, so care should be taken while using PGR, but they are since they are used in very small quantity, they are generally not toxic. okay, but they can affect somehow, like still they can affect if they are continually, if a person who is applying it, or if the plants, if there is continual spray of these PGRs or more pronounced spray, that is if you are using it in a larger quantity also, so it can affect the growth of the plant, it will, or the health of the plant as well as the applicant who is applying it. PGR have an excellent safety record. However, poisoning can occur in plant, animal or humans if wrong concentration is used. Yes, if prescribed dosage is not used, if larger quantities are used, so they may have a toxic effect on both on all plant, animal who consume these plants, and humans, those who consume the plant, or the person who is applying this regulators. Then safety equipment, if they are not used properly, so they may be inhaled, like the person who is applying, he may inhale these PGRs. These are synthetically made chemicals only, so they can affect adversely. Then application times are not correct. If application time is not correct, then again it can have a harmful effect on the health of the plant. Okay, so suppose ethylene. So ethylene should be applied during ripening period or after maturity. If it is applied earlier, so your fruit will not grow any further, growth will be retarded. Again, if ethylene is applied during growth period of the plant itself, so all the leaves will fall out of the plant. Again, your photosynthesis will not be accurate, and that will cause an adverse effect on the health of the plant or the growth of the plant, right? So that was all in this lecture. I hope you enjoyed this lecture. Thank you all.